Hey, Mark Alvarez here, and I have one question for you. Has this ever happened to you? You're just sitting there trying to pet your cute cat you've had for a year, and then it just kind of starts biting all over. Ow, and it hurts. And then, what do you know? Look at that. Sometimes you even start bleeding. That's definitely real blood. And sometimes when this happens, I start thinking, man, how do these wounds heal? How does my finger stop itself from bleeding?
Well, wonder no longer. Today, Mark Alvarez, Nick Warner, and Bradley Turner are going to take you through the process of wound healing. We're talking strictly physical wounds here, not any mental wounds like the one you got when you saw my pathetic attempt at a mustache. Yikes. Regardless, wound healing is a four-step process. The first is hemostasis, the second is inflammation, the third is proliferation, and the fourth is maturation. Nick will take it from here to tell you about hemostasis.
When looking at these molecules, however, you would immediately find a problem. These structures are humongous. Most of them are actually represented as stringy lines or weird paper-looking structures, especially proteins, but that's just not a good way to understand the process for our purposes. So we need to look at these specific regions of these molecules and identify their purposes within a blood clot system. One of the most important molecules we're going to look at in this process are called integrins, which are long receptor molecules sitting on the edge of these platelets. Their purpose is to facilitate interactions between the platelets and other structures depending on the integrin. And in terms of structures, integrins are actually very similar to proteins in the sense that they are amino acids chained together, but the ways these are chained are a bit different. Integrins are composed of alpha and beta subunits, both formed of amino acids but not directly connected by a covalent bond. These subunits are actually usually held together by either dipole-dipole interactions or hydrogen bonding. Now, while there's many integrins that do exist even within the blood system, there's a couple integrins that are more important to highlight for the purpose of coagulation. One being the alpha-2 beta-1 integrin, as it is the structure allowing platelets to bind onto collagen. Now, I haven't mentioned that word yet, collagen. The word is actually Greek for glue, and collagen itself is actually pretty important due to its role as a glue. It has a triple helix structure, with each strand being a polypeptide chain of over 1,000 amino acids. The amino acid sequence itself is pretty common, usually being glycine, proline, hydroxyproline, but those last two can be exchanged for lysine and hydroxylysine. These hydroxyl versions are just simply oxidized versions of the standard amino acids, so they just have an extra alcohol group on them. Now, the reason we call collagen glue is because collagen acts as the base of the clot that forms due to its ability to glue platelets to the collagen, giving strong structural support to the clot. We see this interesting strong support due to the cross-link of covalent bonds between individual collagen molecules involving the lysine and hydroxylysine side chains. And an interesting thing to note about collagen is that we actually see an example of how something we take in every day is so important. Vitamin C is actually responsible of turning lysine into hydroxylysine. With a severe vitamin C deficiency, this cannot be produced, and the chains, the triple helix structure, gets significantly weakened and weakens your blood vessels, which is not good for you. So the moral of the story is, go drink some orange juice. The other big integrin we need to consider is alpha-2b beta-3 integrin. This is one of the more common ones, with about 100,000 molecules of it on the surface of the platelet, and it has the ability of allowing platelets to come and stack together, increasing the size of the clot. And this actually takes place right after the platelet binds to the collagen, forming the base. And we haven't talked much about the mechanisms of the integrins and why they're so selective, but before going further on alpha-2b beta-3, it is a good time to talk about that. Overall, the reason why these integrins are not coming together all the time and linking with other molecules is because they're deactivated. And there's a couple ways this can happen. This can either be the shape of the protein isn't right, so the spot that the interaction takes place at isn't accessible, or there could be a molecule missing that's key to making the interaction happen. Most of the time, these integrins are deactivated because of the lack of a divalent cation. A divalent cation are cations such as magnesium ions or calcium ions. They're actually pretty prevalent in your body and can be released at will by certain cells. Now, once an integrin gets the magnesium or calcium bound onto it, then usually its shape can change, or it now has the correct charge to be able to do whatever interaction it needs. And in terms of what the actual interactions that are binding ligands together, it's usually hydrogen bonds, dipoles, and other types of intermolecular interactions, because these are strong enough to hold, but not so strong that they're unable to be destroyed at the end. And to go further into how important and effective these ions are, it's a good idea to look at the product QuickClot. QuickClot is essentially just gauze, but it contains a special calcium-carrying clay that is ready to release its ions into an aqueous environment, such as the human body. So when the gauze is put onto a very large open wound, something like a gunshot, the calcium ions are released and surround the area of the wound with all those platelets and activate the binding integrins on them, speeding up the clotting process. Now, some clotting factors simply just bind to the integrins like before, but some of the more interesting ones come from those factors that bind to the membrane of the platelet. Those that bind to the membrane itself actually have an interesting mechanism we can discuss. It starts with the platelet's membrane, which is similar to a phospholipid bilayer you'd see in a normal cell. This phospholipid, in this case, is phosphatidylserine, and just like all phospholipids, there's a polar end with a negative charge. This site is a great spot for some of these calcium ions to bind, but the negative charge on the phospholipid is not enough to completely negate the positive charge, and this process allows multiple different factors to be bound if they undergo carboxylation.
As said before, there are 4 steps to the wound healing process. The second step being inflammation. Even though it is considered the second stage, it begins right after the wound occurs. The inflammation process creates swelling and helps to prevent infection. Platelets from blood clots help constrict the blood after injury. Also, white blood cells arrive at the wound site to help start the cleaning process and start killing bacteria. These enzymes carry the white blood cells that create the pain and redness that is usually felt during the wound healing process. During the inflammation step, injured blood vessels leak a fluid called exudate, which contains a high amount of protein. Exudate is made up of serum, fiber, and white blood cells and caused by inflammation only. The serum in the exudate carries all the protein that does not contribute to blood clotting, but instead contains electrolytes, antibodies, and hormones. Both fibrinous and serous exudate fluid are leaked out during the inflammation stage. Fibrin plays a key part in the creation of blood clots to prevent blood loss and to help start the repair process. The fibrin itself contains fibrinogen, which is made up of 3 polypeptide chains labeled alpha, beta, and gamma. The chains are long, elongated structures of about 40 to 45 nanometers in length. These chains are joined by disulfide bonds, which are the coupling of 2 thiol groups with a bond length of 2.5 angstrom. These polypeptide chains have molecular masses of around 50,000, so it is hard to look at the molecule components very closely. However, we do know that some portions of the chains contain carbohydrates which normally have a hydrogen to oxygen ratio of 2 to 1. Mark in the next video will explain more in detail how the enzymes for the blood clotting and inflammation process work.
So by this point, the wound has been sealed and blood will not flow out, but there still remains the problem of the injured tissue that must be repaired. This harkens the beginning of the proliferation stage, where new tissue is grown and new blood vessels are created in a process called angiogenesis. Angiogenesis is when new blood vessels form from old ones. This process is begun when a growth factor called vascular endothelium growth factor, VEGF, is released by endothelial cells in response to being injured. This is just one example of signaling pathways, which are the defining characteristic in regulating nearly every biological process in all organisms. How they work is as follows. In response to some sort of stimuli, in this case wounded cells, a signaling molecule is released. This molecule, in this case the growth hormone VEGF, travels through the bloodstream to its destination, in this case any nearby vascular cells. Now here's the interesting part. The signaling molecule, now called a ligand, attaches to the receptor molecule on the cell of interest, which causes a change in the structure of that molecule, which activates a response mechanism, generally a protein of some sort. But there are more complex systems in which the activated receptor molecule initiates its own signaling pathway, which starts the process all over again. The attachment of ligand and receptor molecule is particularly interesting because it changes the effects of the molecule. That is, the ligand induces a change to the structure of the receptor.
In a way, that can be likened to a catalyzed isomerization reaction. It's not quite the same thing, but thinking of it like this can help our organic chemistry minds understand it better. The next step in proliferation is cells called fibroblasts enter the wound. These fibroblasts insert collagen into the injured site, the same molecule that Nick was talking about before, the one that acts like glue. The fibroblasts create a large patch of collagen that acts as a sort of faux skin. This skin is called granulation tissue and consists of many different things. If you've had a bad cut before, you've seen this. It looks like a flat patch of bloody skin, but don't worry. This just means your body is working and it doesn't even bleed easily. Finally, epithelial cells join together over the granulation tissue, marking the end of the proliferation stage. The next step is called maturation. In this stage, the temporary cells must be removed so the wound can fully close. In order to do this, the collagen changes form. Collagen has many forms, but it switches between two during wound healing. Originally, it is type 1, and at the end, it is type 3. There is one critical difference between the structures of type 1 and type 3 collagen, and it lies in the disulfide knot at the end of the type 3 collagen. This knot serves to strengthen and stabilize the collagen by preventing it from moving. This is needed because it is only after the collagen is converted that they can cross-link and strengthen the new skin. This process begins about 21 days after the injury and can continue for up to a year. After all is said and done, the new skin can be up to 80% as strong as before. It's not perfect, but it's a passing grade.